Hello, everyone. I'm Santiago Callegari. I'm a TA1, and today I'm going to present to you the relevance and the importance of the study population in clinical research. So first of all, assume that you're working at a pharmaceutical company and you're interested in knowing if a drug X will work in patients with hypertension. Then, what are the main considerations to define your study population? Are you going to test your drug in everyone? Or are you going to test your drug in a handful of patients? And if it is this way, which patients are you going to choose? So as you can see, it's not an easy task to define what study population are you going to test in your specific scenario. So why is it important to define a study population? This is because we want to know first our target population is. We want to make an impact in that target population. If you're testing a drug for hypertension, then we know we're targeting hypertensive patients. So if we choose a population that it's not representative of the target population, so that means the whole patients with hypertension, then we are not testing the drug in the population that we wanted to, and we are not improving the outcomes or not improving the outcomes depending on the study on the population we want to address. So that's why it's so important to specify and clearly target your target population and clearly know your target population. Of this target population, you will rarely have access to the whole population. You probably have access to a very much smaller part that is the accessible population. So for example, if you work at a hospital, it's impossible to know all the hypertensive patients, for example, in the United States or in Colombia or in whatever country you are in right now. It's very likely you're in a hospital or in a healthcare setting, and you know a handful of patients that attend to the hospital and are accessible in the hospital. However, not all the patients that go in the hospital will be in your study because of a number of reasons. You may have exclusion criteria. You may not want patients that are very sick. For example, if a hypertensive patient is having sepsis, for example, in the ICU, you maybe are not interested in that specific patient. So that patient is excluded, even if it's accessible. And so on with different exclusion criteria or patients that just don't want to participate in the study. And then at the end, you will have your study population. That is the population that you are going to test your intervention or to analyze your observation. And this population should try to reflect as much as possible the target population. And this is, for example, what has happened in many different articles. For example, in here, when they ask if empagliflozin, that it's a new drug, will work in different types of populations. For example, in here, patients with chronic kidney disease, and in here, patients with heart failure with preserved ejection fraction. And why is it so important to define your population? Because you have two things here. You have the external validity and the internal validity almost collide. Because if you include everyone, if you include all the patients with hypertension, then your external validity and your internal validity actually can be strong enough because you have all the patients. If you have all the patients, then obviously the intervention you are testing will work in all the patients or will not work in all the patients. Even though we don't know the outcome, what we know is that it's going to be representative in the study if the study is done properly. On the other hand, we have the internal validity, and the internal validity can be a good internal validity. For example, if we just test hypertension in females older between 40 and 50 or so with no kidney disease, for example, but the intervention actually doesn't work in males, then we have a low external validity, but probably good internal validity, and the intervention will work in this population. So you can see that external validity and internal validity kind of collide in terms of what your study population are choosing, and that's why it's so important, because you need to define a population that has a good external validity, so you have an intervention that works for your target population, but you also need a strong internal validity, so you know that your intervention is valid and your study protocol is valid within your population. And this is, for example, how specific you want to be. So for example, in this trial, they assessed the sarcomere protein gene mutations in hypertrophic cardiomyopathy of the elderly. So as you can see, you just can be really focused and create populations that are very detailed and very strict. Of course, this limits some of the external validity. And this leads to the sampling. So the sampling, there are different types of samplings: the probability and the non-probability sampling. Sampling is the process by which you enroll patients in your study. Using the probability sampling, and although I will not dive into all the details, what you need to know is the probability sampling means that all the subjects have the same chance of being included in the study, and the non-probability, this is not the case. So there are subjects that have a higher chance of being included. The probability sampling is incredibly rare in medical practice. Why? Because you will need to have using our example of the hypertensive patients, you will need to have a list of all the hypertensive patients in your country or in your population, and then choose some of them to participate in the study. It's much more common to use convenience sampling that is highlighted here. That is the one that is commonly used almost in all the clinical trials. That is just the patients that are enrolled in the study or the patients that attend the healthcare settings. And this has its advantages and disadvantages, but this is a common source of bias for many trials, and you always need to take into account that convenience sampling, even though it's very frequently used, can introduce bias into the study. And just remember, the common sources of sampling bias are when only volunteers participate in the study, the convenience of selecting the sample, the lack of blinding during the sampling process, and the poor allocation in study groups. And that's it, and hope you have a great day.